Hey, good morning, good afternoon, everyone. And thank you, everyone, for attending this month's Jousse Lecture. This is our first attempt at a hybrid format. So if you're experiencing any technical issues online or even in person, I apologize as we try and work these bugs out. It's my delight to welcome Dr. Zariffa today, who will be presenting on a very interesting topic. Dr. Zariffa is a senior scientist at Kite Research Institute at Toronto Rehab, and is an associate professor at the Institute of Biomedical Engineering at the University of Toronto. His research interests focuses on neuroprosthetics and technology for upper limb neurorehabilitation, and it encompasses work in wearable sensors, neural interfaces, electrophysiology, and machine learning. Recently, Dr. Zariffa's team was awarded the grand prize in the Spinal Cord Rehab Innovation Challenge. And this was a national competition that focused on new tools to measure rehab activities and their impact. The title of his talk today is AI-Driven Frontiers in Assessment and Restoration of Motor Function After SCI. If you have any questions in person, I would ask that you hold off until the question and answer period afterwards. If you are online, please feel free to put your question on the chat, and I'll accumulate all the questions to ask him after he has completed his talk. So without further ado, Dr. Zariffa.
All right, hello everybody. Good afternoon. It's really a pleasure to have a chance to be here with you today. Thank you for coming. Thank you, Brian. Thank you, Brian, for organizing everything so well and with lots of technical proficiency, because hybrid meetings are not easy. So I hope everything works. All right, so hi everybody. So my name is Jose. I'm excited to share some of our work with you today. A lot of what we do in our lab deals with measuring function, finding new ways to measure function in the context of rehabilitation problems. And so today I'm going to talk about a little bit the role of technology, the roles of technology can play in the assessment of function, and what we can do with that in the context of assessing and restoring function after spinal cord injury. All right, so I'll start by thanking all of the amazing trainees in the lab that have moved a lot of this work forward, as well as our the financial support from many of the agencies listed at the bottom, as well as of all the individuals with spinal cord injury who have participated in our studies and helped us to advance this work. All right, so the overarching motivation for a lot of our work is the incredible variability that exists in the process of neurorehabilitation. So for two different people, depending on the location and severity of injury, the symptoms and impairments might be different. We can add to that issues of demographics like age and comorbidities. Even if all the biological factors are the same, rehab is about getting people to go back to doing the things they want to do, right? Achieving their goals, and those goals might be different from one person to another. And so the rehab process is usually tailored to what a person is trying to work towards. So that's another source of variability. Between different healthcare systems or different institutions, the amount of time spent in rehab might be different, inpatient versus outpatient, number of hours, technology available. All these things are also adding variability in the process. And all these things being equal, two therapists might also just choose to treat the same patient a little bit differently based on their clinical judgment and schools of thought. So highly heterogeneous process, which leads to the question, how do we know what is the best approach for a specific person in a specific situation? And so that's not a new question. Recently, the term precision rehabilitation has been used a little bit for this, to kind of the idea that for each person, we need to have specific rules that tell us for this particular situation, this is the best approach. How do we optimize the outcomes for each person, considering all these sources of variability? So that's kind of a motivation for our work. So we're interested in developing tools that can help us to measure human function, because in order to make decisions about what is the best approach for this person and for this situation, we need to have data that allows us to have, first of all, evidence-based so that we understand these relationships, and then that allows us to make decisions in the face of a specific person. Okay, so we're interested as biomedical engineers to develop tools to measure human function. And this can take several forms. Some of our work is in wearable technology, which enables us to measure things that we wouldn't normally have access to. We would do work in neuroprosthetics, which are forms of assistive technology that interface directly with the nervous system. And in that context, being able to measure the activity of the nervous system can be used to regulate the behavior of the device or the function of the device. So again, measurement is very important. We also do work with biomarkers, which is the idea of trying to find physiological variables that we can measure that can enable us to predict what whether a specific person might respond to a particular intervention or what intervention might be the best choice for that person. So can't talk about everything today, obviously, but I'm going to talk about two different projects today. And I want to specifically split the time because we often tend to, share the same information with a specific group of people. And sometimes we don't always know what we're all doing. And so I wanted to deliberately kind of go broad and talk about a couple of different things to have more ideas that we can discuss and maybe things that some of you may have heard some of these, but not others. So I'm going to talk about some of our work in wearable technology and some of our work in neuroprosthetics. I'm happy to take questions about any of these topics in the questions. So on the wearable technology side, we've done a lot of work on developing a wearable system to measure hand use at home. Okay. Before I get into hand use specifically, I think it's worth stopping and taking a minute to think about why we might want to use technology to measure function in the first place, right? Why would we use sensors for assessment? So this is kind of a mini SWOT analysis to think about the considerations there. So if we look at the advantages of doing that, strengths, a big one to me is enabling us to measure information at times and in places that we otherwise wouldn't have access to. So we can measure things in new environments, we can measure things at more time points and get a more detailed picture of recovery. So that's not so much about doing things a little bit better or a little bit more precisely, but about doing things that we just couldn't do before, right? So giving us new capabilities. Using sensors can also provide us with continuous values and maybe more objective assessments, which in some cases can lead to more sensitive assessments. Although in many cases, our clinical tools already have good reliability. So to me, really the main advantage is measuring things that we couldn't measure before. What's the new things that we can do? There, of course, are some challenges. These include complexity. As soon as you're integrating new tools, new equipment, that's going to cause logistical difficulties and increased cost, of course. In the context of wearables specifically, there's compliance issues. So if somebody is going off in the community and using the device, you're not observing them directly, which is kind of the point, but at the same time, you may not be sure that they're wearing the devices correctly, doing the procedures correctly, and so on and so forth. There's opportunities in using technology to measure function in rehab. Multimodal, by that I mean that we can have different sensors measuring different things and have a more comprehensive picture of what's going on with a specific individual, because we can complement functional testing with electrophysiology, with behavior in the community, and so on and so forth. We've just put into place an electronic health record. We have Epic now. That's an opportunity to have digital data feeding directly into a central repository, which will enable that information to be collated and presented in a way that can impact clinical decision-making. Obviously, a bit more complicated than that made it sound, but in terms of administrative issues, but it's a major potential of technology that we have to try to exploit. And the other one is really scale, right? Collecting data in rehab is difficult when you have to do manual assessments, which is what we could mainly do now. So you might get one data point every few weeks because it takes half an hour to do the assessment and it's hard to find the time, right? So there's a major bottleneck in how much data we can have about the process based on the fact that we have to manually collect, right? So through technology, we might open the doors to having much larger scale data collection, which will enable us to have this evidence base that can help us to better understand this very heterogeneous process that we were talking about. And then threats, so validation, all the technology that we develop has to be carefully proven to be giving us correct information, of course. In some situations, different technologies seem like they're doing the same thing. There's, for example, many accelerometers on the market. We have to understand what the differences are and whether they're equivalent or not, whether we can merge data from different studies or not, right? And sometimes there's transparency issues. For example, like a Fitbit might give you a number, but there's a mysterious algorithm that computes that number and you don't necessarily know what it is. Okay, so I see these things as kind of background setup for everybody to keep in mind. And now let's dig a little bit more deeper in this issue of hand function that I wanted to present today. So this is the International Classification of Functioning Disability and Health from the World Health Organization. It's a way for us to categorize what we mean by human function. What does the word function mean? We can break it down into three main categories. Body functions and structures refers to impairment, organ level impairment. My muscle is paralyzed. Activity is my ability to perform a functional task. Right? And participation is, and then I use that function to go into the community, go to school, go to work, go to dinner with friends, and so on. So today I'm going to focus on activity, which is, again, can I perform a functional task or not? And the ICF model breaks this down further into two different concepts, capacity and performance. So capacity is somebody's maximal ability to do that task, that function. So under standardized conditions, they're trying hard or concentrating, can they achieve this? What is the best that they can do? Performance is what people actually do in their daily life, right? They're at home, they're busy, what's actually happening? Okay, so capacity is can they do it, whereas performance is do they actually do it? Okay, and so the problem with our current set of assessments for hand function is that most of them measure capacity. They're in the clinic, we're observing the person, we're getting them to do specific tasks, but we don't have any good direct methods to assess performance, what's actually happening out there in the community. And so that was a gap that we were interested in addressing. So we started playing around with a technology called egocentric video. So this is video from wearable cameras. So the person is wearing a camera on their head and it's recording what they're doing with their hands. And the reason we're interested in this technology is that it's giving information about both the person's movements and their hands and function, but also the functional context. It's showing you the task, the objects that they're manipulating. So there's a lot of functional information. It's very, very rich data. At the same time, because it's so complex, we need methods to simplify it and extract information out of it. And so we were interested in combining egocentric video with artificial intelligence methods, machine learning techniques to go through these videos and automatically extract pieces of information that would be clinically relevant. That's a study from a few years ago that was in a lab and just kind of showing this concept of having these algorithms that go and try to figure out what's going on in the video. So the study I want to tell you more about today was conducted by Dr. Andrea Bendini. And we wanted to validate that we could extract from these videos useful information about how much somebody was using their hand at home, and that this information correlated with clinical assessments. Okay, so we want to validate metrics describing the amount of hand use at home obtained by applying deep machine learning methods to egocentric video. So we had 20 people with a cervical STI who took part in this particular study. And so I won't go into details about the inclusion-exclusion criteria, but basically we recruited people who had impaired but not completely absent hand function. So you could tell that their grasp was not, what an able-bodied person might use, but they were still able to do some stuff with their hand. There was something to observe. Demographics are there, mostly male, mostly less severe injuries, mostly HDs, and mostly traumatic. But there was a range of function. Okay, so what did the study look like? So in first session, they came into the lab. We assessed them in person using the grasp and the skim. So grasp being gold standard for hand function, and the skim being also gold standard for independence in functional activities. And then we trained them to use the equipment. We taught them how to use the cameras. We went through that with them. And then we had a conversation with them where we asked them what their regular routine was. What does a typical week look like for them? And then through that conversations, we agreed on what they would record. And so basically we were looking for them to record real portions of their routine that were actually representative of their daily lives, and it still involves some kind of hand function. So we would not ask them to record specific tasks. We would agree on things like, okay, record your meal time on a weekday, your chores on the weekend, and so on. So then they went home, they recorded about 3 to 4 hours of video over a couple of weeks of their real routine. Right? So you use GoPro cameras. Then they came home, so they came back to the lab, they gave us back the video. We interviewed them to understand their perspectives on using egocentric video in the context of rehab for things like privacy considerations and so on. That I'm not going to talk about so much today, but it's published, and if anybody's interested, I can talk about it during the questions. So today I want to focus on the information that we could extract from the videos and whether that was valid information for reflecting amount of hand use. So this required 2 steps. The first one was to develop algorithms that could process the videos in an automated manner, right? So what we specifically were looking for in this case was detecting functional interactions of the hand with objects in the environment. When is the hand interacting an object? When is it not? So just 0 or 1. Okay. And then once we had that information, we wanted to validate the metrics that we extracted from that against the clinical gold standards, so the GRASP, the SCIM, the UEMS. So this is an illustration of that. So we had input videos, and then the algorithms had to do 2 things. They have to find the hands, which is not necessarily easy for a computer to do because the hands are changing shape all the time. They're moving quickly. They're hiding behind each other and behind objects. And then for each hand that was detected, we had to determine whether or not it was engaging in an interaction. So again, 0 or 1. So we get kind of a timeline for each of the 2 hands. And then the metrics that we used to summarize this information were the following. We had percentage, so of the total recording time, how much of it was spent in interactions. Duration was the average duration of individual interactions, and number was the number of discrete interactions normalized to the length of the video. This is some examples of the data. I'm not sure how well you can see it, but basically you can see the variability in these kind of real world situations, a lot of clutter, a lot of very different objects, very different activities, very different lighting conditions. So really kind of messy but interesting type of data set. I won't go too much detail about the machine learning aspects, but basically we compared a number of algorithms to figure out which one worked best. The winner was the one listed at the bottom there, if anybody's interested. And so on average, so the best algorithm had an F1 score of 0.8, and F1 score is a measure of classification accuracy that's between 0 and 1. 0.8 was good, was sufficient for our purposes. We picked this algorithm and we applied it to all of our data. I'm going to show you results in 2 batches. The first one is correlations of the egocentric video measures against unilateral hand function scores. So unilateral UEMS and GRASP scores, which assess hands one at a time. Okay? So a couple of trends to take away from this. There's a lot of information, but one, we had stronger correlations for the non-dominant hand, which suggests that the dominant, because of the nature of the fact that we were asking to do some kind of activities, the dominant hand was often doing something at some point, whereas the non-dominant hand tended to be used only when it had a certain amount of function, right? So if they had more functional non-dominant hand, they were more likely to integrate it into their activities. The other thing to note is that the percentage and number of interactions was better correlated with the duration of the interactions. Duration tended to be a less useful metric in our results. The second set of results is comparing the egocentric video metrics to bilateral measures. So now this is the bilateral UEMS and the SCIM scores. Okay. Again, so on the left-hand column there, we have a combined metric for both hands. And so that's kind of our main measure. And it correlated with UEMS and the total SCIM score. So that was validating the fact that these measures are providing real information. A few other points to note here. High correlation of the number of interactions with the SCIM mobility score, which suggests that people who had better mobility were going around their house and doing more stuff. So that kind of makes sense. For the looking at unilateral egocentric measures, the non-dominant hand again was more useful. Not more useful, but more strongly correlated. But again, percentage and number of interactions were more useful than duration. Okay, so what do we take away from all of this? Basically, the takeaway is that the automated analysis of egocentric video provides valid measures of hand use at home in the case of cervical spinal cord injury. And this is important because it addresses a gap in our set of available outcome measures. We do not have a measure that could objectively measure performance. And now this can do that. So that was the takeaway there. We're continuing to work on this to look at things like reliability and responsiveness to fully validate the measure as something that can be used in clinical studies to help us gauge the real impact of new interventions. Now, I just told you all about amount of hand use, right? But one of the main advantages of egocentric video is that you have a lot of information about exactly how somebody is using their hand. So we want to go beyond amount of hand use and look at quality of hand use as well. And so we've recently been doing analysis of the types of grasps that people are using. This slide is all from manual annotations from machine learning, but basically what we went through the slide, the data, and annotated 4 grasp types: power, precision, intermediate, and non-prehensile grasps. And we demonstrated that the types of grasps that people are using correlate with their level of function. Right? That demonstrates that the type of grasp is an important metric in terms of assessing somebody's upper limb function at home. These are the specific correlations. This is work done by Jousse T. And so basically we see, for example, that people who have higher levels of function will tend to use more power grasps. Somebody who has lower levels of function will tend to use more non-prehensile grasps, where they're kind of pushing or holding without using their fingers. So now the next step for this is to try to automate this. And so Mehdi is working hard on developing algorithms that can be used to automatically classify grasps in egocentric video. One study, for example, where we demonstrated that we can detect the use of a tenodesis grasp, which is a common compensatory posture. And now we're working on identifying power grasp, precision grasp, intermediate grasp automatically. Okay, and lastly, both of those topics relate to basically using this as a research tool. We want to be able to have measures that enable us to look at the effectiveness of new interventions. But the other question that comes up is this: is there any role for this in clinical decision? If you give a therapist information, direct information about what's happening with their patient at home, would that make any difference to the care that's being delivered or to the choices that they're making? That's the next thing that we would like to understand. And so we're currently running a study on that here at Lyndhurst. So if anybody is or knows outpatient OTs who might be interested, we'd be happy to talk to them. What we're doing is we're recruiting patients, therapists and their patients, right? So the patient goes home, records egocentric video at home, brings it back. We create a report of that video, give it to the therapist, and then we discuss with the therapist how this information relates to their decision-making, how useful would it be, in what way would it be useful to them, and so on. So that's still ongoing, and we hope to have results for that in the next few months. Okay. So as I mentioned in the beginning, I really wanted to have like 2 completely different gears to just vary it up a little bit and give everybody kind of a sense of the breadth of research that happens at Kite in SCI area. So for the second half of the talk, I want to tell you about implantable closed-loop neuroprosthetic devices. So what do I mean by that? Okay. So this falls under the context of FES, or functional electrical stimulation, which is a technology that we can use to cause paralyzed muscles to move through electrical stimulation. And so for many years, there have been systems developed that can be implanted in the body and can trigger movement of a person who's paralyzed through a specific pattern of electrical stimulation. Challenges of that is that if you just have a fixed pattern that you always trigger, the movement is always the same and is not adaptable. It cannot deal with it with any unexpected event, like the hand hit a wall and isn't where you think it is, or the muscle is fatigued. So that's not at all how the human body controls movement. We're constantly integrating sensory information in order to make motor decisions. So the sensory motor loop that's always regulating our motion. So in order for this implantable FES business to get closer to natural fluid movements that a human would do, we need to have a sensory aspect to that system. What we're wanting to do here is extract sensory information that's already flowing in the peripheral nervous system and use that as a feedback mechanism for the FES. Here's a couple of examples from old studies. The one on the left here It's a situation where this was a human study where they had an implantable grasping FES system. So they would close the hand to enable somebody to hold an object. And what the sensory portion of that was that there was a, they were recording the activity of afferent nerves in the hand and detecting when an object was slipping, detecting information from the sensory receptors on the palm. And if the object was slipping, they would grasp harder, right, to kind of ensure that this is a stable grasp. On the right is an animal study where we had, it's not our study, but they had a cat with spinal cord injury on a treadmill, and the FES was being used to move the cat's legs and make it walk on the treadmill. Now, they were recording activity from the peripheral nervous system, specifically the dorsal root ganglion, and decoding the kinematics of the legs. They were decoding what the joint angles were of the different parts of the leg, and then based on that, deciding how they were going to stimulate to cause a specific gait movement, gait pattern. Okay, so these are both examples of using information from the peripheral nervous system to control FES. So we're interested in moving this forward and improving the performance. And so we're interested in a problem called selective recording. So what do I mean by selective recording? So on the left here, we have a nerve. This is a human median nerve. And so towards the top, it would be going towards the spinal cord. And then as it goes away from the spinal cord, it's progressively branching out and sending branches to different limbs and organs. Okay? And so each of these branches is going to have a different functional meaning. So one branch will be carrying motor commands to a specific muscle or bringing back proprioceptive information from a specific joint. Okay? And so I would like to be able to monitor all of that, what is happening in all these different branches. So I have two options. I could either do what's shown on the right there with the little red stars. I could just go and implant lots of devices in different parts of the body that measure what's going on in different places. That's going to be problematic because that's going to be too much hardware. The complexity of the implanted system is going to be too high. What I would like to be able to do instead is do what's shown in the green electrode at the top there on the top right, is to have an electrode that's on the main trunk of the nerve high up close to the spinal cord, but that's able to differentiate all the signals passing through at that level. So whatever signal passes through the nerve where the green electrode is there, it's going to say, okay, well, this is a signal coming from that branch or going to that branch over there, right? So to be able to discriminate the activity recorded at that level. And so that's why I'm saying selective recording. We want to be able to record and then figure out where that signal is coming from within the nerve. Okay. All right, so what are the challenges involved in doing that? So this is the peripheral nerve. It's basically a bundle of nerve fibers. So tens of thousands of nerve fibers packed into a space a few millimeters or maybe a couple centimeters long in diameter. And so what you would like to do in this case is go and monitor the activity of small groups of nerve fibers within this nerve that carry the information that we're interested in. So from an engineering perspective, that's a challenge because it's a very tightly packed structure, and we're targeting something very small and precise. So people have approached this in different ways. Here's a couple of different categories of devices. On the right is intraneural electrodes. So these are electrodes that you implant inside the nerve. There's going to be hardware going in the nerve tissue itself. And so there's electrical contacts that are placed close to the axons, and so can record the activity of very specific groups of axons. The challenge is, again, you're putting stuff in the nerve, so in terms of long-term safety, that may not be desirable or ideal. We're not necessarily sure. Category is extraneural electrodes, and these don't penetrate the nerve, they wrap around the nerve. They're sitting on the surface of the nerve, okay? And so these are very safe. They've been used in humans for periods of 15, 20 years. We know they work, they're clinically suitable, they're fine. The problem is because you're not inside the nerve, you're on the surface, the signal that you're recording is smaller, noisier, and less selective. It's less specific to specific groups of axons. So what we would like to have ideally is something that's up there on the diagram, something that's very selective, but also very safe, right? And that produces no damage to the neural tissue. So the question we were interested in was whether we could take, these extraneural electrodes and increase their selectivity so that we could have an interface that's both selective and minimally invasive. So how do we get there, a big part of the idea is to use nerve cuff electrodes that have many recording contacts, that have grids of electrical contacts around the nerve. And so the idea is that as neural activity flows through the nerve, there's going to be electric fields that happen around the surface of the nerve. And the patterns of variations in those fields that you can detect by having these grids of measurements will be a little bit distinctive, and we will be able to use those variations to separate activity coming from different places. So how do we, what kind of experiment do we have to do to test this? So this is a nerve cuff recording, and a recording from an electrode that's wrapped around the nerve. You can see they're very small. This is microvolts, and they're very noisy. Okay, this is a nice recording from the standards of nerve cuff recording, right? So what people might typically do is they would take a chunk of the signal and average it to get kind of a global measure that was a little bit cleaner, but not very specific. We wanted to go further. We said, okay, if we look at a very specific narrow window and we pick a specific spike, okay, we want to take each individual spike that we detect in these recordings, and we want to classify. We want to say where this particular spike came from. Which branch of the nerve was this spike coming from? So that's what we're going to do. Discriminate the individual naturally evoked spikes recorded from different neural pathways using multi-channel nerve electrodes. And so, a couple of notes. I say by spike here, we're talking about the activity of a small group of nerve fibers that are firing together synchronously. And I say naturally evoked spikes because sometimes these studies are done by stimulating a nerve electrically, and then you get this really artificial and really big signal, but that's not what physiologically happens in the body. So we want to use real spikes from just real movements and real sensations that were natural kinds of activities. So we want to classify each of these spikes. And so if we can do that, I say in the title, we can increase the temporal resolution because now we're classifying spike by spike instead of a large chunk of signal. We can increase the spatial resolution because we're figuring out where in the nerve each one is coming from. How do we do this? This was work done by Ryan Coe. And so the idea was to create these spatial temporal signatures. So this on the left here, we have a nerve with a multi-channel nerve cuff placed on it. So it's a grid of recording contacts. If you look at one of these contacts over time, you're going to get a signal like you see at the top there, where it's an actual potential shape. But if you look at a specific time instant and you look at the whole map, you see the spatial variation at the bottom left. If you put these two things together, you get what's on the right. So a spatial temporal signature, this map where each row is one contact and each column is all the contacts at one time instant. So basically, the concept was that different neural pathways would produce slightly different signatures. So these signatures could function as kind of like a fingerprint that is specific to a particular pathway in the nerve, right? So signals coming from different branches in the nerve would have slightly different signatures that we could use to classify where a spike was coming from. So this was a preclinical study. So we had 9 rats. We implanted the 7x8 grids of these electrodes with 7x8 grids of contacts on the sciatic nerve. And then we had 3 different types of stimuli. So the concept of the experiment is that there's different types of stimuli that are known to activate different branches in the nerve. So if I dorsiflex the ankle of the rat, I know that's going to activate this action potentials in a specific fascicle, a tibial fascicle. If I plantar flex, that's going to activate activity in a different fascicle. And if I poke the heel, that's going to activate activity in a third fascicle. So based on what stimulus I'm applying at any given time, I know where the activity is in the nerve. And so the goal is to take the recordings from the nerve cuff and correctly identify where it was coming from. So I know the answer because based on what I'm applying, the nerve cuff doesn't know, it just has to predict which type of stimulus this was. Okay, I'm not going to go into the signal processing, but basically we have a series of steps that are going to clean up the signal a little bit, help us extract specific spikes. So if you look at the bottom there, so each of these blue bursts here has many spikes in it, and every time it goes above a threshold, we extract spike. This is a spike seen in one channel, but again, we have a grid of these channels. We have 56 of them. So this is what it looks like altogether. Or I can take this information, I could represent this exact same data in a different form. This is, again, a spatial temporal signature that is characteristic, hoping, of a specific, the activity in a specific branch. So the question was, can we take these maps and then use an algorithm to determine where this one came from? That's where we turn to machine learning. We use a technique called, or a type of algorithm called convolutional neural networks, which are just very common neural networks that are specifically good at classifying images, right? So if you go on the internet and you want to learn about deep learning, you'll probably see an example where they're classifying dogs and cats, that kind of stuff. So they're good at specifically dealing with images. So we use that type of technique to process these spatial temporal signatures. And so we've got these three variations of this kind of image that we can get, and we're trying to classify it. Okay. So again, the three classes, so based on these three stimuli: dorsiflexion, plantarflexion, and pricking. And again, we know which one is correct based on video recordings of when we were applying the stimulus. We use the CNN to predict. This is the structure of the neural network for people who are interested in these things. Each of the two branches here is a very simple conventional neural network, CNN. And then there's two branches because based on how you order your contacts in your map, you get slightly different shapes. We weren't necessarily sure which one was going to be the most useful. So we tried both. It turns out the combination is better than either of them alone. So we have two branches and we're feeding information together. And so based on these signatures, it's going to go and classify which branch of the nerve the activity came from. And this turned out to work fairly well. So the last line there is this algorithm I just showed you. We got 80% classification accuracy on these three class problems. So this was the first demonstration that it was possible to classify individual spikes from outside the nerve. Right, that hadn't been done before, so we're pretty happy with that result because it really suggests there's a possibility to use these types of electrodes that are very safe to get very precise information about what's going on inside the nerve. The next step was we just wanted to confirm that this information we were decoding was actually physiologically meaningful. So we wanted to predict a specific physiological thing that made sense, a variable that made sense. So we focused on the angle of the ankle joint, so decoding proprioceptive information. So what you're seeing on the right there, so the top is the actual angle of the ankle. The second row is one channel of our recordings. Again, there's actually 56 of these. And then the next two lines are the outputs of the algorithm. So what it's doing now is it's predicting the firing times of two different pathways in the nerve, the dorsiflexion and plantarflexion pathways. And then we smooth those out, so we get these firing rates at the bottom. And then what we did was we fed these firing rates to another neural network whose job it was to predict the angle based on this information. Here are some results from that. And so this is one good result and one less good result. But what you're seeing here is the black line is the real angle, it's based on video annotations, and the red line is the predicted angle from the neural recordings. And you can see on the left, there's a very good match. The right is an example of one that didn't work as well, but over our 9 rats, the correlation between these two lines was 0.82, so demonstrating good performance in tracking the physiological variable. Okay, so when I started talking about this in the beginning, I mentioned closed loop FES, right? So everything I just told you about was offline. So we had a rat, we collected data, we went back to the lab, Ryan worked hard for months, and then it was offline analysis, and we got some results. What we want is to be able to do this in the loop, that we want to be able to decoding information in the nerve, and then right away use that to control FES delivery. So the next step was to try to do that. So in this set of experiments, we had a rat, again, with a nerve cuff on its sciatic nerve. Here, data is being recorded from the nerve cuff, decoded using the type of machine learning algorithm I just explained to you. And then we have rules that say, depending on what was measured from the decoded nerve activity, we're going to stimulate the nerve through the same nerve cuff. So the same nerve cuff is alternating stimulating and recording. And it is going to cause specific movements depending on what information was decoded from the sensory activity. Okay, so this is work that Eugene Huang did. And the the rules were fairly simple. So this was just kind of a state-based control system. So what it was supposed to do was the following. So at first it's at rest, nothing is happening. If we prick the heel, that's going to cause it to start moving. And at first it's going to dorsiflex the ankle. And then if it detects it's in dorsiflex state, it's going to plantarflex. And if it detects that it's in plantarflex state, it's going to dorsiflex. So you're going to get this oscillation. Okay? But it's not a pre-programmed oscillation. All we've programmed in are these rules. Start if you detect this, change states if you detect that. Right? So I'm going to show you an example of this in practice. It's an animal experiment with a little bit of blood. If that disturbs you, please look away. It's about a minute long. But I think it's worth showing. So you're going to see the pricking at the beginning. That's going to start the ankle moving. Once again, one nerve cuff that's doing both detecting and stimulation. And so you can see the oscillatory movement that happens. And again, this is just based on detected nerve activity and a set of rules. And from one nerve cuff, that's both stimulating, and between bursts of stimulation, it's recording and feeding that information to the neural network. Okay, so an example of a simple closed loop FES system in practice. This is just some group data, so from different rats. So each row here is a different rat, and just showing how many trials this worked. So a trial is a set of oscillations, and for each one, how long it kept going before it made a mistake. We also tried a slightly more complex set of rules at the end, but basically showing that it worked across many rats, but there are some reliability issues. Wasn't always very robust. Sometimes we have to do a lot of tweaking. And so the next steps are definitely to improve the robustness and the reliability of the methods, because these are, again, very noisy, very small recordings. And so it's difficult. And we're trying to pick up really subtle patterns in these neural recordings. So feasibility is demonstrated, but reliability still has to be improved. Okay, so the conclusion from this part is that classifying individual naturally evoked spikes from a nerve cuff recording is possible. Okay, again, that's important because it's about both spatial and temporal resolution. We can decode things from a safe electrode that doesn't penetrate the nerve, with individual spikes with very high temporal resolution, and be able to sort of separate the activity coming from different places in the nerve. Okay, so this gives us basically a channel of communication between the human body and a piece of assistive technology that enables that more detailed interaction that will make it possible to have hopefully these closed loop systems that provide more natural and fluid movement that's a bit more similar to what our natural bodies can do. Going forward, we still need to look at improving performance in chronic implantations. So this was just an acute experiment. We implant it and see over the course of several weeks how performance changes. We need to look at more complex nerves. This was a rat sciatic nerve, which is a very common model for these types of studies because it's small and simple and convenient to access. But a human nerve is going to have a lot more fascicles and going to be larger, it's going to be more complex. And so we need to understand how these techniques work in the context of more complex anatomies and more complex patterns of activation. So those are things that we're excited to be continuing to work on. Okay, so I'll tell you a couple of very different things, but just to summarize a little bit, or some outlooks. So takeaway messages, I think, here is that the reason we're interested in using technology for assessment and measurement in rehab is that it provides opportunities to access previously inaccessible information, right? So again, the main message, it's not about doing something a little bit better, it's about doing something that we couldn't do before. And so, but as that happens, now we have more sources of data, bigger data sets, more frequency of data collection, we end up with tons of data, right? And so that's where we need AI techniques to help us manage large, complex types of data and be able to tease out those really non-obvious relationships, right? And then allow us to move towards this concept of precision rehabilitation, where we can really predict the optimal approach for each given individual based on a set of, based on being able to accurately track their status and their function. Okay. That sounds great. Obstacles to doing this are, so there's definitely scientific obstacles, but there's also a lot of systemic obstacles. which tend to be the first ones that we encounter. Anybody who's ever tried to do a large study in rehab will know that this is difficult because you need to usually get multiple centers to collaborate together. That can cause logistical difficulties. You have to work with institutions to overcome a number of barriers. And then even once you've collected data and come to conclusions and have demonstrated that something works, integrating new technologies into healthcare environments and healthcare systems is also a challenge due to a variety of administrative, financial, and regulatory considerations. Right. And so I don't want to end on a downer note, but the point here is in order to move things forward, we need to be aware of this, and we need to engage a number of stakeholders or interest groups early on and regularly in order to be able to manage these obstacles as things move forward, not to spend a lot of time developing something that then gets stuck on translation. I'll make a plug for something that Chris Musselman did a few years ago. Very minor for me was we did this slightly modified version of an existing framework called the FAME framework, which stands for feasibility, appropriateness, meaningfulness, efficacy, and economic evidence. And this is basically a framework that helps us to think about the different pieces of the puzzle that need to be in place for successful translation to occur. And our modification here was about making it into a staged process where it's overwhelming when you're first starting and you have a fresh new idea to have to deal with everything at the same time. So obviously things are going to happen progressively, but as the technology or the intervention develops, there's different people that have to come to the table and whose input is needed in order to support successful translation. So going from clinicians and patients to hospital administrators to economists who can help us understand those aspects of the question. So as things move forward, we really need to think outside the strict box of the science and have those relationships in place that can help us to overcome the systemic obstacles that are inevitably going to be something that have to be dealt with in any kind of translational endeavor. Okay. So I'm going to stop on that note. Thank you. I hope I left lots of time for questions. And thank you very much for your attention. It's a pleasure to be here.
Hey, thank you, Dr. Zariffa. And that was like our first live clap in years at the Jousse Lectures, I think. But thank you. I appreciated how you were able to take like very difficult concepts and explain it in a manner that even I could understand. So that's quite an accomplishment. So I open it to any questions that we may have, people who are here in person and even online. If you have a question online, please put it in the chat or raise your hand and you can ask your questions. So are there any questions here?
I'll repeat the questions up for online.
Okay. So on the topic of selectivity of recording for implantable cuff electrodes. So I know like one major issue that isn't necessarily addressed yet is the topic of the movement of the electrode after implantation. So what are your thoughts about dealing with that problem? Because is it like an issue of just we need a better algorithm with more data to be invariant to this kind of movement of the electrode, or are there other possible solutions?
Thanks, Nick. I'll just repeat the question. The question was about how robust decoding of nerve cuff signals might be when the electrodes are moving over time on the nerve. So great question. I think it's going to be a combination of factors. So as I mentioned, robustness is definitely the thing that needs to be improved. And there's, I think, unexciting and exciting things that need to happen at the same time. So like just really good mechanical design to anchor the electrode and make sure it doesn't move too much, and like suturing it to the nearby tissue and like this kind of stuff is not super exciting, but it's actually really, really helpful in making sure that things are stable over time. So there's that kind of mechanical design of the implant is a big part of it. But then we're very interested in developing algorithms that are robust to these types of things. So we've done some simulations, for example, to show how do things change over time as a result of movements? How does that affect the performance of neural networks? I'm interested in looking at developing neural networks that are adaptive over time and that can adjust their behavior, and what are different strategies to do that, to make sure that they can kind of learn on their own over time how the signals are changing and try to adapt to things over time. So I think there's some of those kind of exciting high-tech approaches, but also some of just the basic stuff of just making sure that it's not moving more than it needs to, and that's. Thanks so much.
So we have a question in the chat.
Okay, so why do you anticipate the future of this field of research will look like? And if you, sorry, what do you anticipate the future of this field of research will look like? And if you were to estimate, when would you start to see this commonly used in clinical practice? Okay, well, I think obviously the stuff I spoke about in terms of human studies with the egocentric video stuff is much closer to translation, I hope, than the implantable neuroprosthetic systems. For the egocentric video side, we very deliberately made sure to use devices that are off the shelf. You can go to Best Buy and you can buy one of these cameras. Anybody can get one very easily. It's not something special that we built that we have to start a company and so on and so forth. So all the complexity is just software, which is easy to share, right? So it's algorithms and software. So that was the reason we did it that way. So I hope that this is something that people can start building into their studies now. If somebody has an upper limb intervention that they're wanting to try, like send people home with the camera, get some data. We can start to have data that validates that supports the use of these techniques. So that I think is fairly close to translation, at least translation as a research tool, which was its original kind of goal. In terms of clinical adoption, again, we're working with therapists now to understand what role it might play, but then we get into issues of how does that information get integrated into the EHR, which is something that again requires ongoing discussions to deal with. In terms of the implantable stuff, of course, that translation is longer. But again, part of the reason that we're interested in nerve cuffs, and like the easier way to be selective is to plug something into the nerve, right? The reason we're interested in nerve cuffs is that they have been used in humans for very long periods of time. We know they're safe. So if we can get, again, the algorithms and the software and using the AI possibilities to increase the amount of information that we can decode from hardware that exists, then we have a leg up from a translational perspective because, again, the devices are already things that have been proven to be safe. Next one.
When you were looking at the previous that you showed less than as the algorithm collected more data, did you see the transition from sensory input to motor output increase the speed, or is that something you find?
Yeah, thank you. So just to repeat for the people online, the question was about for the closed loop FES experiments, as more data was being collected, did the interactions or the efficacy of the sensory to motor decision making change, I guess. So no, in the sense that these were short experiments. So there wasn't like long-term opportunity for long-term changes. Also, these are rat-specific models. So like the machine learning system that you train on one rat is specific to that rat because it depends on how the anatomy of the nerve is slightly different for everybody, and exactly where the electrode is positioned is slightly different. So you can't transfer that from one electrode from one experiment to the next experiment, right? So and since they're fairly short, there wasn't really time for that adaptation to happen. It's an interesting question. If it was implanted for a long period of time, would you start to see things like that happen? I don't know. Yeah.
I think it's very interesting, fascinating that you can come up with a proof of concept and show that spatial temporal signature is feasible. But that was in a very highly controlled environment, very specific to those three tasks. I was just wondering what your strategies would be in terms of improving the reliability in a context where tasks are so much more variable and in a clinical population that is as variable as spinal cord injury.
Yeah, no, for sure. The great question. That's what we're excited. We want to move forward to do chronic implantation studies because a lot of these questions, I mean, I can talk about it, but we won't really know until we do it, right? But in terms of the summit strategies, some of it is based on existing technologies like muscle activity will interfere with nerve recordings, but people have 30 years of experience in figuring out strategies to prevent that and mitigate it. So there's some issues. In some places we can build on existing technologies. In terms of the translation to people with spinal cord injury, again, these are very individual neural network models. So it's really about finding a calibration process that makes sense. When some of our computational modeling, we demonstrated that you can, the algorithm can't completely learn on its own over time, but it can kind of slow the rate at which you have to recalibrate. So we can have kind of a hybrid model where you can slow down the rate of the decline of the performance, but once in a while you have to recalibrate. So if this was a person with a spinal cord injury with an implant, there would be some calibration procedure where they would have to do some specific procedure. And then the network would know, okay, this is what the signal looks like right now or today, and then it can adapt. So there's going to be some kind of combination of technologies that are robust to disturbances or changes over time, and just the realities of having to sometimes recalibrate systems. That kind of practical thing.
Okay, so I'd like us all to thank Dr. Zariffa once again for a wonderful presentation. Next week, we will be on break, so there won't be a Jousse lecture next month. Instead, during this time, there will actually be residents' virtual best practice rounds, the presentation by Dion Diep. The following month, we will return, and the guest speaker at that time will be Susan Ascari, who will be talking about mindfulness and spinal cord treatments and spinal cord injury. If you have any topics or speakers you're interested in, please feel free to contact me. If this is your first time attending Jousse lectures and you would like to be on the mailing list for future presentations, please email me as well. So thank you, everyone, and have a great day. Thank you once again, Dr. Zariffa, for a great presentation. Thank you.